Hello, everybody. My name is Courtney Martin, and today I'm going to be talking about bioweapons. Now, I'm going to be talking more about what kind of pathogens can be bioweapons and what can make a good bioweapon, and not necessarily bioweapons in general. So, in this presentation, I'm going to talk a little bit about what bioweapons can be. And then I'm going to talk about the body's immune system because of the defenses that we have and how those defenses can be um, hindered by pathogens. Then I'm going to be talking about virulence, and we'll get more into what that is and how that can help pathogens. Then we're going to talk about pathogenesis and examples of bioweapons. So just a little bit about what a bioweapon could be. It could be microorganisms such as viruses, bacteria, fungi, and protozoa that can be used deliberately in order to cause large populations to become diseased or dead. And these microorganisms are going to use your cells as hosts to replicate and spread, because that's what pathogens are going to do to survive. And in the process, they're going to damage our host cells, causing an immune response, and this is where we're going to see physical symptoms arise. And examples of bioweapons that we'll be talking about today are going to be smallpox and anthrax. So before we can talk about pathogens, we're going to talk about our own body's immune system and how it would react to pathogens entering our system. So in our bodies, we have three lines of defense. We have our natural immunity, adaptive immunity, and innate immunity. So with our natural immunity, it's going to be our first line of defense, and normal examples of that would be our skin, our hair, our tears, among other things. And some pathogens can be stopped from affecting you just simply because they don't have access to your body in that way. However, they still can get access to you if you were to cut your skin, or if you were to inhale it, or if you were to eat it. However, with that being said, going on to our second line of defense would be our innate immunity. So with our innate immunity, it's going to be a non-specific defense inside our body. So by non-specific, I simply mean that the cells that are used in the innate immunity second line of defense are going to be cells that basically kill any cell that is identified as non-self. So that's going to be any type of foreign molecule that your body sees as a threat. So the cells that are usually going to be seen in our innate immunity are going to be our inflammatory response cells, our natural killer cells, and our phagocytic cells. So with the inflammatory response cells, those are just going to cause an increase in blood flow to the area of infection, which is where we're going to see more basophils, neutrophils, things like that are going to help get rid of that pathogen. And that's also why usually when we are infected in that area, you're going to see a lot of swelling, redness, and it feeling a little more throbby because there is more blood flow going to that area. With our natural killer cells, they're basically exactly their name. They target and kill pathogens, or in this case, non-self organisms in the body. And then lastly, with the phagocytic cells, those cells are going to basically engulf any type of non-self cells that they might find as a threat and basically kill them. And yeah. So then going on from our innate immunity, we have our last line of defense, which is our adaptive immunity. And this is where we're going to see our T cells, B cells, and other antigen presenting cells, which are just further going to rid our body of pathogens. So the difference between our innate immunity and our adaptive immunity is that our adaptive immunity is going to be more from memory. That's where you see our antigens, which come specifically from pathogens. In that way, our body can identify exactly what pathogen has entered our body, and what T cells and B cells can be used to better get rid of the pathogen. Okay, so now after talking about our body's immune system, we're going to go on and talk about virulence and pathogenesis. So I'm going to go to the virulence slide first, just so it'll make more sense. So with virulence, we're talking about how well a microbe can infect a host. So if a pathogen has a higher virulence, that means that more people are going to be infected and have the disease and be affected by that disease, whereas a pathogen with a lower virulence is going to have a harder time with being successful in infecting a host. And in addition to virulence, we're going to have tools that are used to aid in pathogen infection and invasion, which are called virulence factors. So basically, those are things that are going to better help us, or not us, pathogens infect us. So some examples of virulence factors are capsules, which are going to be like the main structure, kind of like a cell wall or cell membrane, which are going to help stop phagocytosis in our second defense of our immunity. And then we're also going to have pili, which help with movement and also adhere to pathogens and host cells, and also flagella, which also just helps with movement. And I'll talk a little bit more about how that helps pathogens better infect us. So going back to our pathogenesis slide, I'm going to talk a little bit about the steps of pathogenesis. So pathogenesis is basically how a pathogen can be successful in infecting a host, and it's going to have kind of a direct correlation with high virulence. So if a pathogen has high virulence, it's going to be really good at pathogenesis. So with the steps of pathogenesis, we have number one, which is entry, which is basically what I talked about earlier about how a pathogen can get into our system in the first place. And that could be from a cut on the skin, through the nose, through the eyes, or through the mouth, among other ways. Number two, you have tissue attachment and colonization. So this is where we can see our virulence factors coming into effect. With tissue attachment, we have those pili, which are going to help attach to hosts. So the pathogen is able to use that host cell as needed and replicate. Number three, we have immune avoidance, which is where we can see the capsule coming in, as well as the flagella. The flagella will help more in two as well, due to movement. So the pathogen will be able to move throughout the body and infect more cells. But the capsule will be more helpful in step three with immune avoidance because it is helping to avoid phagocytosis. Number four, we have host damage, which is just cell death, which is going to occur after pathogens have infected the cell, replicated, and things like that. Going on to step five, you have pathogen exit or pathogen spread. So depending on what pathogen it is, it can exit basically the same way it enters, and can be spread through the air or through bodily fluids, and there are also other ways that it can happen. So moving on to more specific examples, we have anthrax and smallpox. So with anthrax, we know that in 2001, we had an anthrax attack soon after 9/11. Fortunately enough, only 22 people were infected and only 5 died. We were a little bit more prepared, obviously, because this was a newer thing that happened. But it is still very scary because, especially with anthrax, it is naturally occurring. So people don't necessarily have to find it in a lab. And it can be very versatile because it can be put into food, water, it can be turned into a powder and put in envelopes like what happened here, et cetera, et cetera. With anthrax, it is also basically tasteless, odorless, and microscopic, so it's very hard to detect if you're not really looking for it. So you never know when it could, be around you. So with anthrax, it does have virulence factors, including protective antigens, lethal factors, edema factors, and capsules, which are just going to help with killing the host cells, with swelling, and inhibiting phagocytosis. with smallpox, we have an attack that happened back in the 1700s with the colonization of the United States. And this is where we see the British give blankets infested with smallpox to Native Americans, Native Americans. But unfortunately, because of this, the Native Americans basically didn't stand a chance because they didn't have their first line of defense, their second line of defense, or their third line of defense. And I say this because the virulence factors in smallpox, they both are basically going to interrupt the innate immunity in the communication and signaling process. So it's going to take your body longer to even realize that it's been infected. So with all three defenses down, because Native Americans don't have a third line of defense because they've never seen smallpox before, so they have no memory of it. This is why we see basically a large deficit in Native Americans. With that being said, your body has a built-in system of how it defends itself. However, that defense can be fooled and unfortunately can lead to death. Pathogens have been designed to take over a host cell in order to survive, and being able to manipulate pathogens to give them an advantage with virulence factors can be very, very deadly. And this is why I think that bioweapons are very scary. Pathogens are very scary because there's a lot of unknown, and you never know when something bad could happen. Thank you for watching. Thank you for listening. And I hope you really enjoyed my presentation.